Well, first, my thanks to the organisers for inviting me to speak to you. I'm somewhat daunted and feel very small in relation to the list of speakers we heard from your president yesterday. Nevertheless, I'm going to talk to you about the most important process on the planet, and that is photosynthesis, the source of all of our food, the source of the oxygen we breathe, and it'll echo quite a bit of what Pierre has just presented to you. So this is where I work most of the time, the Institute for Genomic Biology at the University of Illinois. It has all of the gene editing tools you've been hearing about, sequencing tools and so on for dealing with DNA and molecular biology. But right in front of it is the field of maize, and this is in fact a 150-year-old experiment looking at how maize treated in different ways affects the soil. So outside of an experiment at Rothamsted in England, this is the oldest experiment in the world on sustainability. And it's very important to the state of Illinois. Illinois produces over 10% of the maize produced in the world, over 10% of the soya produced in the world. Illinois is about the size of Italy, and during the summer, about 70% of its land surface is covered with either maize or soybean. So although we're in an institute for genomic biology, it's also very connected with the land and with farms. Another very important resource to me at the University of Illinois is the National Center for Supercomputer Applications. This looks like a building, but actually it is the largest public domain computer in the world, with many thousands of cores in there, and it's very important to the computational side of the work I'll present to you. I also have a small appointment at the University of Lancaster in England, which is connected to our sustainability interests. And you may have heard about recent changes in the politics in the United States. So the University of Oxford has given me refuge for 1 year as a sabbatical visitor to get used to those changes in politics in the United States. What I want to present to you really is in 5 parts. Why we have world food insecurity emerging, which may sound strange at a time when food prices are very low in real terms to us. Why is photosynthesis important to this? Mathematical modeling to show the way on how we can modify photosynthesis. And then I'll give you an example of proof of concept from modeling through to a more productive crop. And finally, come back to the issue of bioengineering crops, where really Europe has been the center of opposition to these changes. So, rising world food insecurity. If we look at what we've achieved over the last 50 years, we've really seen an almost linear increase in the yield of rice and wheat, also maize and soybean, in terms of yield, in terms of tons per hectare. And if we project that forward, we could say, well, if we can continue doing this, what we could achieve by 2050. The problem is that the United Nations Food and Agricultural Organization, other commentators project that by 2050, we're going to need a higher increase than we're achieving at the present time. And that is challenging because actually if we dig deeper into these numbers, what we can see, this is the case of rice. Indonesia, India and China are the world's biggest producers of rice. Rice is the largest single vegetable source of human calories in the world. What we can see is that during the 70s and 80s, they were able to increase the yield per hectare of land by almost 30% per decade. By the first decade of this century, that increase had come down to single figures. What is alarming about this is that in the first decade of this century, China was investing 100 times more than it was in the 80s in improving rice yields. This tells us it's getting harder and harder to get these increases. Now, food insecurity is a serious business. Most of the changes in the politics of the world result from food shortages. As Louis XVI could tell you, he lost his head over the fact that there was insufficient bread for the population of Paris. That led to the French Revolution. More recently, in this UNFAO report that's just been published, where UNFAO actually point out that after a period in which food security has been improving, we're now moving into a situation where it's getting worse again. And what they point out is all of these red lines show major food riots in different countries. And those coincide with periods over the last few years when food price indexes have risen. So the Arab Spring, all of the turmoil that has produced, is closely associated with food riots resulting from increasing prices. Of course, as we heard in the last talk, we could obtain more food by bringing more land into agricultural production. But this is poorer land than we're using today. It's environmentally very susceptible. Much of it is the forests that we really need to keep intact for biodiversity, the Amazon, and so on. So what this means is we need to achieve more yield on the land we're already using. Now, another problem is we don't just need more yield. What we pointed out in this study was that, for example, the Midwest or the Corn Belt of the United States provides about a third of global corn production, about a third of global soybean production. The yields have steadily increased, but what we point out in this study is that quite soon we're going to run up against a barrier, because although we've been increasing yields, we haven't been improving the efficiency with which the crop uses water. So although most of this land is not irrigated today, we will only be able to get more yield by irrigating more and more of that land. And already, agriculture is the biggest consumer of water on the planet. So it's competing with many other, if you like, uses of water, including our drinking water. So we need to increase yield per unit area of land, but we also need to do it sustainably. So why is photosynthesis a part of a possible solution? Well, if we consider what determines the genetic potential of a crop at a given location, i.e. this is growing with the water, the nutrients, the agronomy it needs to fulfill its potential, it's determined by the amount of solar energy, if you like, visible sunlight, because that's what photosynthesis uses, available over the growing season, how good the crop is at capturing that solar energy, how good it is at converting that captured solar energy into biomass, i.e. converting light energy into chemical energy, how good it is then at partitioning that biomass into the part of the crop we care about, the grain of rice, for example. Okay, so if we look at a modern soybean crop growing on our university farm, what we see is that the efficiency, interception efficiency, is remarkably high. About 89% of the visible radiation is captured. If we look at partitioning, how much ends up in the seed of soybean? 60%. If we're going to have some stem and pod to hold that seed, we're obviously not going to get much higher than 60%. So if we look at pre-green revolution, i.e. 1960s cultivars, we find that their interception efficiency was about 60%, their conversion efficiency way down about 15%, and then their partitioning efficiency about 30%. After the Green Revolution, these things were improved. The Green Revolution identified ways to do that. And really, we're now hitting the biological limits of what we improved in the Green Revolution. One thing that the Green Revolution did very little for was conversion efficiency. And conversion efficiency is basically photosynthesis, the process by which plants convert sunlight energy into chemical energy. Now, a question would be, if we improve photosynthesis in a crop, can we really get a higher yield? And this is something that has been doubted for a long time. One of the things I was able to set up when I first moved to Illinois were these experiments where within our crop we put in rings, and working with the engineers, what these rings do is as the wind is moving in a certain direction, on the upwind side we add carbon dioxide, and of course the higher the wind speed the more carbon dioxide we use, so that within these rings we're elevating carbon dioxide to a constant level. And what we were targeting was the carbon dioxide level we think we will have in the year 2050 to find out what that would do with crops. Carbon dioxide is a limiting substrate for photosynthesis. So if you elevate it, you get more photosynthesis, and what we saw was a 25% increase in photosynthesis. And that translated into about a 16% increase in soybean yield, a 12% increase in rice yield, and a 15% increase in wheat yield. So we know if we could boost photosynthesis, we could get more yield. Many people have questioned, well, surely evolution will already have optimized photosynthesis. But I'd argue against that because, first of all, selection in the wild is for fecundity. Getting your genes into the next generation, how you do it doesn't matter. It's being able to do that. And we have many minute plants in the wild which are highly successful at doing this, but very unproductive. Our crop ancestors evolved in a carbon dioxide concentration of about 200 parts per million. Today, the carbon dioxide concentration outside is over 400 parts per million. Half of that rise has occurred just in the last 50 years. That's far too short a time for evolution to adapt. Also, what you can see here on the left are ancestors of soybean and of wheat. These are plants growing in very open habitats where all of their leaves will be in sunlight. Today we grow our crops in these dense monocultures where we may have 6 layers of leaves, and only 5 of those are in the shade. So we've taken sun-adapted plants and made them largely shade plants. And again, this is relatively recent that we've done this. Evolution has not had time to adapt. So why do we think we could do it today? Photosynthesis is the most studied topic in plants, and it's very uniform across plants. We pretty well know all of the steps involved. We know all the genes underlying those. We know the kinetics of each step of the process. So we can simulate that in high-performance computing. When we predict what we should be changing, we then have crop transformation, genetic transformation, is becoming increasingly routine. So I want to start this by looking at mathematical modeling. The reason we do this is, for example, systems analysis has become very important in industry. if you think of a car production line, you might have 1,000 workers on that line. Where do I put my workers to maximize productivity? And of course, there are a lot of permutations of that, but a computer can work through those permutations very rapidly. We heard yesterday about Watson, for example, being able to predict things faster than the human brain could. And that's exactly what systems analysis does on a production line. So what we set out to do was apply the same thing to the process of photosynthesis. And what you can see on the right is, you probably can't read all of these steps, but all of the 160 discrete steps of photosynthesis. So what we set out to do was to mathematically model that. And on the left, you can see a picture of rocket. And what I want to point out to you is that plant biology can be rocket science, because we had our system of 160 differential equations that we wanted to numerically integrate. This was particularly difficult because some processes operate at nanoseconds, others at minutes. And we were struggling with that. And a rocket scientist, Eric de Sturla, said, I have exactly the same mathematical problem with balancing rocket motors, and this was my solution. We applied that to our system, and it worked. Of course, the second problem, can we get it to run? So, a second problem is that, of course, a leaf is not in the same environment all of the time. It's moving into shade. It's try again. No, it's not going. Well, if we could see this, what it would show is a model simulation of a soybean crop growing, so that, you know, what we're doing with this is we're working through trying to optimize our photosynthetic system to the average conditions under which the crop grows. I think we should probably move on and abandon that. What happened there? It's okay, you can see the simulation. So we're basically trying to integrate our biochemical simulation into then a simulation of the canopy. And what this actually does is it takes the light conditions over the day, but I'm not showing that because you'd see a series of flashes. And so we grow the crop through to maturity, and we try and optimize the system to that. Now, of course, these models can look great, and we can sit at our computer all day, and you know, a model looks like that. But now, when you try and implement this into a muddy field situation, the picture can look a lot more ugly. But nevertheless, the Gates Foundation were reading our results from computer simulations, and eventually they said, well, we're interested in this, and we're willing to put our money where your mouth is, and you show us you can really do this in practice. So we took up the challenge, and this is the team of people who got together with me to go where no sensible plant scientist has been before. This is Martin Parry from the University of Lancaster on the left, Don Ort from the United States Department of Agriculture, and then on the right, Murray Badger from the Australian National University, and Christine Rains from the University of Essex in the UK. And we set out to try and do this, and our approach is, as illustrated here, we use the mathematical model and billions of permutations, as we heard very much yesterday with Watson, to really then try and distill out what are our best bets? And then we try to engineer those into a crop. Now this sounds strange because our crop is tobacco. Tobacco, that's not a very important food crop. Why do we use it? Well, tobacco is very easy to engineer, but it is a crop. It produces a canopy like soybean and wheat and so on. So we can test it in the field situation, and if it works, then we go on to the much harder tasks of putting this into cowpea, which is the most important vegetable protein source in sub-Saharan Africa, into rice, the most important source of human vegetable calories in the world, and into cassava, which we've just heard about. So we've basically identified a series of different ways in which we could modify photosynthesis. Our team's been working on all of these. I'm just going to give you one example, which is really my own laboratory's work. So a proof of concept. When a leaf is in full sunlight and then it goes into the shade, changes occur. A leaf in full sunlight cannot use all of the light it's receiving. And in fact, that light, though, excites the pigment molecules, the chlorophyll, and if that energy is not dissipated, oxidizing radicals are formed, which would literally bleach the leaf, just as if you put a colored garment in sunlight, it bleaches. So the plant deals with this by dissipating that energy as heat. It induces a mechanism to get rid of that excess energy. But when the leaf goes into the shade, say a cloud passes over the sun, it carries on doing that for some time, even though it could now use all of that light in photosynthesis. And it takes some time to get to the state we want, where it uses most of that energy in photosynthesis. So basically what happens is, when the leaf goes into the shade, it's inefficient for a period of many minutes before it regains its efficiency. Now, in a plant canopy, leaves are shading each other, and as the sun crosses the sky, it means points on the leaf are going in and out of shade quite routinely. And this just illustrates to you the real data of a leaf, where 1 minute it's in high light, the next minute it's in low light. We computed from this how much potential photosynthesis is lost by this slow recovery. It's about 20% of possible carbon uptake. So we looked at how could we speed this up genetically. We identified 3 genes that we should upregulate from the modeling work, and we then put that into tobacco. We used a system to try and identify the transformants which were performing best. And what we were able to do was to speed this process up. So, of course, what we really wanted was bigger plants. And here you see 3 different modified plants with upregulation of these 3 genes against WT, which is the wild type, untransformed plant. So it looked great in the greenhouse. We then set about trying this out in the field. This is quite a difficult experiment to do. As you can see, it involves many people setting up the experiment. And then this happened. Oh, and this is what we're looking for. We're looking for a bigger plant. So, and then this happens. This was the highest rainfall ever recorded in late May in Illinois. And it wrecked our experiment because the plants at this end of the field became anoxic, they were starved of oxygen, and it completely wrecked our experiment. So bad things can happen, and they can set your research back a year. All researchers have experienced this, and those of you who are thinking about research, you often have to have some heartbreaks before you actually have some success. But the next year, the experiment worked, and we found very large increases actually by this manipulation. We got a 20% increase in productivity in our best line, 14% in another. So that was a nice payback. Don't know if we can hear this, but this was how the American Association...
They ramped down the efficiency of photosynthesis to avoid damage to key molecules. The drop-off is abrupt. The plants need to protect themselves, but getting back up to speed is slow, like the way your eyes slowly adjust after entering a dark room. It's estimated that 20% of plant productivity is lost to this slow ramp-up in response to rapid changes in light. That's a lot of corn not made. Where does all this variation in light come from anyway? From clouds passing over the sun, from wind blowing leaves around, casting moving shadows from other plants, even their own leaves. In fact, from the point of view of a leaf, even the change in the angle of light as the sun moves through the sky can cause abrupt variations. Researchers wanted to see if they changed this curve, the speed at which a plant returns to full efficiency, if that could have a big impact on productivity. By swapping a few genes from one plant to another, they were able to change photosynthetic recovery time. Plants with these genetic changes recovered about 15% of their productivity, giving them bigger, broader leaves, taller stalks, even more heft in general. Since the mechanisms targeted in this research are common to a large swath of plants, this could be tested in all kinds of crops. For soybean breeders, a 2% increase in yield is huge. So what would a 15% increase in yield give us? Use less land for agriculture? Feed more people? Maybe both.
So they describe, the American Association for the Advancement of Science describes it far better than I can. So we're now, of course, setting about putting this into the crops we care about, and this is cowpea, rice, and cassava. And so we're hoping to see the same results in these. One of the other, of course, issues is that as you're changing many plants, you really want to be able to describe these plants, many hundreds of them, as they're growing. And this is a solution that our agricultural engineers have come out with. What you can see there is a robot steadily moving through our plants, and what it's doing is it's measuring the stem width, it's measuring the height of each of these plants, it's measuring the color of them, it's also using hyperspectral, the reflectance. This shadow you can see in the foreground is a drone which is coordinating with the robot, so it's looking from above as the robot is looking from below. And this is, we've now pretty well able to sequence every one of these plants. This is really helping us now to understand what the genome, all those genomes are telling us, and we can then do large-scale association genetics on that. So finally, of course, what I've told you is really genetic modification in one form or another. We've heard a lot about CRISPR, and that is something where we think there are CRISPR ways where we could get, i.e. DNA editing ways, we could get what I just showed you. But particularly in Europe, we have protests like this. As we heard this morning, these beliefs are so entrenched that whatever data we throw at this, we probably aren't going to change minds. But as Robert Parlag pointed out, the poorest in our world, who probably benefit most from these changes, are really the ones this opposition impacts most. And I'd just like to sort of finish by talking about gene editing, what we can do. One of the big factors improving crops during the 70s and 80s was radiation breeding. This means You take your plant or your seed, you bombard it with ionizing radiation, produce many mutants. Some of those mutants are more productive, but it's very random. You may change one gene that gives you higher yield. You may change 200 other genes and you've no idea what you've done. CRISPR allows us to go in and change that one piece of DNA without anything else. So what we're saying is, this type of behavior is perfectly acceptable, whereas this is not. So, is this all important? You know, for us, food is really very, very inexpensive. Can we possibly need more of it? But keep in mind there are parts of the world where 60–70% of the household budget goes on food. If food prices double, we hardly notice it. For those people, it's a disaster. And this is a paper that Johannes Kromdijk and I wrote pointing out any innovation we make today in crops, as we heard from Pierre in the last presentation, will not be present in farmers' fields at scale for at least 20 years. So although 2050 sounds a long way off to us, it's very important to this chap here, or this lady. I'm not sure whether this is a boy or a girl, whatever. This issue is going to be very important to them. And I'd argue that we owe it to this generation to put these technologies on the shelf so that they are available if they are needed. And indeed, our colleague, TJ Higgins, is working with the Nigerian government on actually getting some of these GM crops available to breeders in Nigeria and other West African countries, and I hope this is how our technology will go. If you want to learn more about what we're doing, we have a Twitter feed and a website. So I hope some of you may explore that. And finally, I should thank the many hundreds of people who've worked with us on this project and made it possible. Particularly thank the Bill and Melinda Gates Foundation for taking a risk with us and funding the bulk of this research. And so I finished with an incomplete list of all the people who have made this work possible. So thank you.